I'm happy to introduce our speaker this morning, who is Dr. Keith March. Dr. March is the director of University of Florida Center for Regenerative Medicine. He's been at that post, I believe, since 2018 when he was recruited from the Indiana University School of Medicine. He's a renowned cardiologist and national leader in regenerative medicine, and he's dedicated his career to bringing new medical approaches to patients. He has more than 150 publications and more than 55 worldwide patents. Notably, he invented the Closer, which is a widely utilized patented suture closure device, which closes a puncture wound in an artery following heart catheterization. And this device has been developed by Abbott and is currently used in the clinic. He served as a scientific advisor to numerous pharmaceutical, biotechnology, and medical device companies, and now they're doing outstanding work at the University of Florida in the area of cardiac regeneration. So with that, I will turn it over to you, Dr. March.
Well, good morning, everyone, and thank you so much for that very kind introduction. I appreciate that, and it's an honor and a pleasure to have been selected to join you today, albeit at a distance, to celebrate the Heart Day, to participate in your celebration of the Heart Research Month and Day. And what I'm going to be talking about today, as it says in the title, is some of the thoughts that we've been evolving over the last couple of decades. We'll be focusing on some cardiac and cardiovascular models, but I'll also bring some other models in. Oftentimes, the cell therapies that we're working on are related predominantly to vascular flow, to ischemia, in fact, and tolerance of the organs to ischemia, as well as to inflammation. And what we've found is, as we think about the cell therapies, we are moving them away from autologous cells towards allogeneic cells, and then eventually towards the secretome of allogeneic cells. And that'll be a theme that I'm going to expand on here in the next few minutes and throughout the talk, that allows us to ultimately look at putting these cell therapies, as it says, in a bottle on the shelf, and eventually even treating organs ex vivo. In other words, moving into much more feasible ways to implement cell-based therapies over time. See about moving the slide. These are my disclosures. A company that I work with on umbilical cell preparations in particular, that we have several clinical trials that are ongoing. I won't be talking about those today, but that, and then Secretome Bio is a small biotech company that we spun out of our lab to really work on bringing secretome of adipose and other mesenchymal cells into patients targeting neuro diseases initially. Again, I won't really be talking too much about that today, but a little bit I will share. So by way of outlining the talk, I'd like to describe our discovery about ASCs, or adipose stem or stromal cells, and their role as paracrine secretory factories. We really began to realize that a lot of what the cells did, by virtue of how they would communicate a therapeutic effect, whether a rescue or a repair effect predominantly, was mediated by what they secreted. And when we first brought that idea forward in 2003 and 2004 in the first publications, we were the first lab to suggest that for adipose stem cells. It was met with editorials that were, although certainly polite, pointed out that we were being very countercultural, because at that point it was thought to be the case that it was necessary that these cells would actually differentiate into something that was not originally what they were, but become some kind of a parenchymal tissue component. While they can do that when in the context of an endothelial partner cell, or an endothelial progenitor, and therefore form vascular networks, and we showed that as well, by all means, that is not the most common way that they can be effective in therapy, but rather it was really what they secreted. And so that, launching of the paracrine hypothesis and describing its evolution will be a theme throughout here. I'll then talk about some of the specific cases where we've used that secretome in various animal models to therapeutic effect. Some of them are listed here. We'll talk through some of those, and then describe the transition as we've begun to test secretome from an in vivo scenario to treating organs, or in fact organoids, in culture, with a target orientation of enhancing organ survival for transplantation. This could apply to a whole range of solid organs, and cardiac in particular has been our focus with my background as a cardiologist and a biochemist. So as you hear about these things, I hope that you in the FIU engineering world will consider if there are ways that we could actually ultimately collaborate. It would certainly be neat to do something that puts together collaborations from our institution to yours, North and South Florida, so to speak. Being that we're discussing this in Florida, I thought I would use a key lime analogy. Basically, the first generation of cell therapy, if you think about it, taking, for example, as we began in 2001 and 2 to take fat, adipose-derived cells, and process the fat enzymatically and mechanically down to these adipose stem or stromal cells, you get a very nice yield, certainly a much higher yield of these mesenchymal cells derived from fat than you do from bone marrow, for example. But really, there's a limited amount. It's autologous, and I think of it very much like a first generation. It is a first generation form of therapy. You're harvesting from your own little key lime tree, if you will, if the key limes are the cells, just enough for you to use. Second generation cell therapy, again, continuing this analogy where the key limes are like the cells, here we take the cells from another individual. For example, their fat, their bone marrow, their umbilical cord, and we plant them, in this case, in a culture dish, in a petri dish, or in hyperstacks, or in bioreactors, depending on the scale. And we plant an orchard from a donated key lime tree, in other words, donated tissue. And then we can have lots of cells that are cryopreserved and, can really treat a whole town of people. And that is, the next stage of cell therapy. The next one, which is really analogous here to the secretome, is you find out what the cells are secreting. You make an extract, if you will, make that key lime filling, and then you can put it in different scenarios. You just have only the filling, or if you're into engineering, into scaffolds or whatever, maybe you put that filling into some sort of a scaffold, in this case, a graham cracker crust, and you make something new that is based on what the cells have secreted. That's third generation. I like to think of it. As you can see, I like food analogies. It's early in the morning, so I like to think about this coffee or key limes. Harvesting from your own key lime tree, then, again, to summarize, that's like an autologous, expanding that tree, expanding it into an orchard, allogeneic. Each cell becomes many, many cells. And then the third generation is collect what those cells secrete and count it. And the nice thing about secretome, not only is it acellular, not that that's critical or important in its own right necessarily, but it does markedly advance the ease of supply chain, because no longer do you have to have the cells as viable cells maintained under liquid nitrogen or under cryogenic circumstances during shipping and entirely until they're used, because the secretome can be maintained under normal storage, freezer conditions, or at room temperature, it appears, or even lyophilized so that they could be reconstituted. So think of the dream of having cellular secretome with its various complex pleiotropic mixtures of growth factors, cytokines, and so forth, as well as extracellular vesicles, available in a vial, in an ambulance, or in the emergency room. I was talking about the morning and coffee, so another analogy, just to drive that a little further home, think about stem cells as like coffee beans. Cell secretome is, making the extract. And that's much more convenient in some ways. So instead of storing or brewing those cells, we bottle the coffee, the secretome, serve up the therapy, leave the cells behind. And that's been a major focus of our laboratory, moving that forward in the past few decades. Now, we began with cells of human. Initially, actually, even earlier, we did some mouse work, but we found that the xenogeneic cells even were well tolerated and had therapeutic effect. And since it would be important to bring the human cells forward for FDA regulatory, obviously you're going to use human cells in a human treatment scenario. One of the first things we asked was whether we could actually salvage ischemic hind limbs by the intramuscular or later intravenous injection of adipose stem cells. So these are the feet of nod-skid mice, which are in particular immunotolerant breed. And in this case, the femoral arteries were ligated entirely, thus to create a peripheral vascular disease model, essentially what people have with dry gangrene. And you can see in the animals that received only medium injection, that is a control or placebo, saline essentially, or media carrier, their feet are very necrotic. They're gangrenous, similar to what happens in the most severe vascular disease. And you have autoamputation, in fact, of the toes. However, in the cases where cells were injected, you have a marked improvement in the viability of the tissue. And while it's not by no means perfect, a much lesser degree of the distal necrosis, gangrene, and amputation. So that was the way we began to study it. And when we look at the blood flow, now this is an intravenous experiment. When we looked at the blood flow, we also saw enhanced revascularization manifested by the relative blood perfusion. If you look at the y-axis here, this looks at the relative perfusion compared to 100% being normal on the contralateral leg, where you have not occluded the blood flow. So compared to that, when you do the surgical ligation, there are still some collaterals, but basically your perfusion measured by a laser Doppler system is about 10% or less compared to the 100% normal on the other leg. Over time, there is very little gradual, but some gradual growth on the leg of animals that did not receive cells. However, those animals that received the injection of ASCs, and notably here intravenously so, had a marked improvement in their relative perfusion, such that it was about 50% recovery by day 10, a very accelerated perfusion. And thinking about that, we recognize there is an important component of tissue rescue, not only of vascularity. We could see that happening microscopically, and it takes time to build vessels. So we went looking for the cells to see where they were. We labeled them with green fluorescent protein and with a lentiviral construct, and then injected them. In this case, they were injected intramuscularly, put them right where the ischemia was. And what we found was that the human ASCs were localized in a perivascular position in post-ischemic skeletal muscle. We're staining for smooth muscle actin here, seen in red. That's the endogenous arterioles. And the cells are just directly adjacent to that, but they do not seem to be turning, we saw, into myotubes, nor are they actually part of the vessel, but they're adjacent to the vessel and adjacent to the muscles. So this began to really, at that point, both confuse and excite us, because we could see that they weren't becoming the parenchyma, but they were doing something of substantive therapeutic potential value, as you just saw in the earlier slides. So we began to wonder whether these cells were actually perhaps acting as supportive neighbor cells, much as glia do for neurons, for example, and secreting these factors. And in fact, this is not meant to have you read or memorize all these factors, but the point is, when we did initial array blots to look at the proteins that were there, we found many, many proteins that the cells were secreting into the conditioned media. And this paracrine factor expression then became what we really started to look into as potentially mediating all of this. This was the first article on the ASCs in particular. Now I'm going to show you a little bit about extending that into the heart realm. We began to look at a similar model, the nude rat heart, again with human ASC grafts by direct injection. And these here we're looking at an antibody-based detection, where the human HLA is detected here in green. And when we find those cells that were adjacent, they were adjacent to the alpha sarcomeric actin of the myocytes, but not the same. So here we fuse the pictures. This is the human cells on the left panel. Next panel over is the rat heart. Then we have DAPI to look at the nuclei, merge them all together. And you see that here in this particular article, and I'm not having time to show that here, but we showed significant improvement in function and diminishment of the size of an infarct in a rat infarct model. And again, this was being mediated by the cells, but they weren't becoming myocardium. In complementary cells, around the same time, we were looking in pigs. We wondered where the cells were going, because as I showed you earlier, we discovered that when we injected such cells intravenously, even though it wasn't in the area of ischemia, for example, the leg, there was significant improvement, very significant. And so how is that happening? Were they homing of necessity or not to the injured tissue? And what we found is, in fact, sometimes that happens to some degree. It does, but not most of the cells. So this is a pig study where we're injecting cells into an area of the heart, and an injured heart, for example. This is the LAD. We're gamma camera imaging the 111 indium labeled cells. That's a typical imaging technique that's actually used typically to watch white cells or neutrophils in humans if you're looking for an inflammatory area. But here we were labeling the ASCs, or in other studies, mononuclear cells. And what we found was that almost all the cells, 90+% of them, trap in the lungs. So these are actually the silhouettes or images, if you will, of the lungs trapping those cells at 1 hour, and that was still true at 24 hours. Eventually they redistribute in traffic, but the fundamental finding here is that the cells are not necessarily where the main injury is, in this instance in the heart, but rather they're trapped somewhere else in the body and effectively begin to act, be hypothesized much like an endocrine organ secreting these factors at a distance. So since then, in our own lab, working with many investigators, both at my prior institution as well as here at UF, and with many other collaborators, both across the country and around the world, we have looked at each one of the disease situations or pathologies identified here in the black around here, and either published article manuscripts or abstracts. And in the cases where it says secretome around the side, those are the ones where we have seen that the secretome emulates the therapeutic benefit of the ASCs themselves. And one might ask, how is it then that so many things can be done by the ASCs as well as the secretome? And I think the answer to that, in part, is shown here. The adipose stem cells we found when we went looking for them, knowing what their surface markers were. Now, this is just a couple of years after that last publication I showed you. We knew about PDGF receptor beta, also known as CD140B, and that particular receptor was a good marker for most of these cells. So we went looking within human fat, and this is a nice image done where CD31, an endothelial marker in particular, is marking the endothelial layer here shown with the adipose microvasculature, and you can see the marking with CD140B of the ASC endogenously sitting there, basically holding on, sort of like, I can't really show it too easily because I'm not there, but holding on to the vessel. You can imagine both a structural or mechanical as well as a chemical, biochemical interaction between the ASC and the endothelial cells. And it turns out that as we extended this observation, Dimitri Traktuev in particular, working with me on so many of these studies over the time period, we found that those cells were not only found in adipose tissue, but basically everywhere throughout the body. And so what we understand now is that the MSCs are essentially in the perivascular or periendothelial, sometimes in a larger vessel, we call it the periadventitial compartment of all blood vessels throughout the body. And so they really act like what I would consider a nice analogy, the repair stations of the body. They're placed on those highways of the body, the blood vessels. And much like repair stations, they don't necessarily move to where the repair needs to happen. They don't come off. The repair station doesn't, move down the highway to your car, but rather what it does is it has toolboxes in it, much like the paracrine factors that are necessary for repair. And, essentially what happens when we take either ASCs or similar MSCs from other sources, as such I mentioned, umbilical cord or bone marrow, and then take those paracrine factors, we're concentrating that toolbox of the body and bringing it to where it might be useful in particular. So this is a little table about the gradual evolution of publications relating to the role of paracrine factors in ASC or MSC stem cell function. And it's been, pretty much exponential growth in that understanding of the field since our first descriptions of it. So what does this secretome contain? It contains multiple physiological activities that are largely therapeutic in complementary ways. So for example, angiogenesis, and this slide was built around in particular some neurovascular findings, but angiogenesis in any area of the body is typically present in these activities of the healthy ASC secretome. I emphasize healthy because if the secretome comes from donors with particular issues, whether smoking that we've published on or aging, senescence, that alters the secretome of these cells, which I think then begins to contribute to negative advantage from the cellular perspective. And so that's where the whole idea of senolytics and senescence begins to emerge. But for the standard, the healthy cells, we're really looking at angiogenic phenotype, pro-angiogenic, pro-neurogenic, anti-apoptotic. So pro-life, anti-death, if you will, and then also inflammation, a decrease in inflammation and immune reactivity, both at the macrophage and at the T cell level, where typically M1 is moved towards an M2 phenotype, for example, using that formalism, and T effector towards a T regulatory as well. And those are some of the key activities of the secretome. Thinking a little bit further, again, a different analogy, when you think about single factor like a growth factor versus cell product therapy, it's really thinking about pharmacology versus physiology. In the first instance, it's reminiscent of one tuba, one particular factor playing very loudly, meaning at a high concentration, and that can make a lot of sound. Or you can have an orchestra where many instruments are playing much more softly individually, but they create a symphonic effect with interacting and reinforcing synergistic sounds, or in the case of growth factors, effects on various receptors, and they still create a very substantial effect by virtue of that complementarity. I'm going to look with you at some more in vivo targets where we actually move from the cells, as I've been showing you here to this point, to the secretome. So one is with regard to stroke. In fact, that was the area where we first began to test this idea. At that time, it was a very untested idea. It was really new and novel. We cultured the cells in a protein-free minimal medium. We harvested the medium, removed the cells and debris, and concentrated it through centricon filters to 50 or 100-fold, and then provided that concentrate into a rat model of stroke, or it could be thought of as cerebral palsy in the neonatal rat, due to Hypoxia-Ischemia. And this was done with Yansheng Du, our neurological neuroscientist collaborator. In this case, much like I showed you before with the legs, the right common carotid was ligated, at this time, of course, supplying the brain, and that causes a hemispheric stroke. You can see the dramatic loss of neurons here in the middle slide compared to the normal structure on a cross-section of the rat brain on the left. So the dramatic loss of neurons in that hypoxic ischemic scenario with neonatal rats. And if we infused the ASC secretome that had been concentrated, as I just described, into the animal intravenously, so intravenously, systemically, even as late as 36 hours after the onset of ischemia, a month later, you have very dramatic protection structurally, which is accompanied by functional therapeutic protection as well, both in areas of memory and motion, motor and sensory. All of those we measured in various ways. Here's a little bit of just a graphic in terms of the protection about neuronal death. This is survival of neurons in the ischemic hemisphere on the hippocampus area. And important to recognize that when we treated, in this case, the code that we were using for the secretome was NFX101. With that, we substantially had an improvement, whether we treated at 24 or 36 hours, in terms of the survival of neurons post-ischemic onset. And I wanted to contrast that here, that 24 to 36 hours. By the way, the effect is lost at 48 hours. So there seems to be a window of opportunity, probably correlated both with cellular death and also with the blood-brain barrier being more permeable and then losing its permeability over time. But that window of opportunity extending for 36 hours is radically different than anything else that we have in medicine today for stroke, because the clot-busting drugs basically have to be provided intravenously or intraarterially by 4 to 6 hours, or else you have too much of a risk of hemorrhage and it's not useful. In other studies, and we've now looked at 3 different laboratories doing similar studies, not our own labs, we don't do neurological models, but we also have seen a decrease in the infarct volume from control as we implement greater and greater amounts of the dose of the adipose-derived secretome, the decrease of the stroke volume, volumetric loss in the brain to as low as 2% compared to the 35% in that hemisphere. That's accompanied by an improvement in the behavioral scores in a dose-dependent way, and in fact, an improvement in percent survival of the animals post-stroke. So we became very excited about that opportunity, and that then prompted us to begin to push towards moving this into a human trial. And right now we're working to build the GMP necessary amount of this kind of secretome to be able to dose in a human scenario. Moving to another area, secretome also we found just recently and published this, reduces inflammation and preserves barrier function in acute lung injury, in this instance caused by lipopolysaccharide or endotoxin. So you see the control lungs, the LPS exposure after 24 hours or 48 hours, you see a progressive inflammatory infiltrate in the lung, in the alveoli. However, co-treatment with ASCs themselves, or at the lowest two panels, ASC conditioned media, markedly inhibits that inflammatory infiltrate. So very important change in barrier function. And just last year, we were able to show that the concentrated secretome of ASCs also limits in a similar way influenza A virus, viral induced lung inflammation. So you see the influenza virus here in the middle panel. I hope you can see the cursor that I'm moving, the mouse. This is a lot of inflammation that is onset due to the virus, whereas in the context of the concentrated secretome, a much lesser degree of inflammation, as well as an improvement in oxygen exchange reflected in the lowest graph here. And so now we have hopes that this could even be useful in other forms of viral lung injury, COVID, for example. Finally, and we don't have time to look at all of the different areas that we've studied, but I'd be happy to share more of that with anyone in the audience who's interested. You know, if you get in contact with me in the future, my email is very simple. It's kmarch, K-M-A-R-C-H, @ufl.edu. So I want to talk now about moving into organs, the heart in particular. Transplant is a very key target for us. We understand that while there is a great demand for cardiac donation, roughly 50,000 people being on the transplant list at any given time to be considered, about 10,000 hearts become available through some donor tragedy every year. And of those, only about 3,000 transplants are conducted yearly. Why is that? Well, about 70% of those hearts designated by donors as available for transplant are not used, and about half of that are not used due to projected time of ischemia prior to the implantation being longer than would be surgically and medically acceptable, by which I mean a sort of maximum ischemic time that people usually use for a donor heart to be devoid of blood flow, meaning the whole heart is having a heart attack, is about 4 hours. And if you go longer than that time from recovery of the heart from the donor to its reimplantation in the recipient, longer than that, you get basically an increasing risk of what's called graft dysfunction. And that graft dysfunction or failure really creates a problem, because then the recipient doesn't have a functioning heart, and you need to go emergently back to either mechanical support or find a new heart, which of course is very difficult in any kind of a rush sort of scenario. So generally speaking, the hearts that are going to be transferred for longer than that period of time, if you consider the recovery surgical aspect and the implantation, you only have 2 to 2.5 hours to transport the heart. So this leads to the discards. So we began to look at the question of whether or not secretome perhaps could in fact be useful in the Langendorff heart preparation ex vivo. And so here what we show is a actual beating heart function ex vivo. This is specifically a rodent heart, and in this case, it's actually beating at several hundred times per minute. You can see that happening there. We'll do it one more time right here. And this allows us to evaluate the developed pressure as well as the heart rate outside of the animal over periods of time. Now, the idea of an increased shelf life, of course, would lead to more than a linear improvement in organ donor utilization potentially, by which I mean if we asked the question, could we perhaps use the secretome to extend that ischemic tolerance time? And if you went from, say, a 2-hour transport to a 4-hour transport, which means a 4-hour to 6-hour shelf life, because that really covers a radius-dependent geography, that would markedly increase the area over which such an organ could be utilized. Thus, the impetus to study this sort of approach. So this, in the Langendorff heart preparation, we're looking here at a rodent heart. You can see the pressure develop, you can see the EKG. And when you measure what's called the rate pressure product, that's the beat rate of the heart, times the developed pressure within the ventricle from systole to diastole. At baseline, you initially get some particular number shown here at the top, labeled baseline. But if you take that heart and then treat it outside of the animal with what's called University of Wisconsin, it's one of several clinically used transport solutions that to some degree limits that deterioration of the heart when it's in the cold. You basically, what's done surgically is the heart is perfused before donation with such a preservative solution and placed at 4 degrees or similarly cold environment. And if you do that for, say, 6 hours, which is what we did here in this experiment in the fridge, then bring it back out, rewarm it, and reanimate it, the heart begins to beat again. But its function is markedly diminished. Look at these dark squares at the bottom. If you do the same thing while co-administering Secretome in that transport solution, we discovered that, in fact, now the heart recovery function was better than halfway back towards normal. So we had a lot of encouragement that, in fact, this was a possibility. So then we worked out the idea of a heart transplant transport model in the laboratory with our wonderful colleague Pinar Zorlatuna at Notre Dame. And she had developed some approaches using both IPS derived from blood cell human cardiomyocytes in a dish, as well as in what's called myocardium on a chip models, which I won't show here today for time, but in both of the simpler models of the cardiomyocytes, as well as the more complex models that incorporate other cells. We then asked the question, could we superfuse these cells with University of Wisconsin or other similar media for 2, 4, or 8 hours, and then look at their recovery in standard media represented here in light brown, either without or with the addition of the conditioned media or secretome. So this is how the cells look beating in a dish. You can see them kind of doing the wave, and these are human heart cells, and of course we can't really study the human hearts that are good hearts ex vivo because those are the ones you need for transplant. So we can't use those very well experimentally, but we can ask the question here about the human biology. And when we did that, we discovered that in fact the adipose stem cell secretome markedly improved rodent heart and human-induced pluripotent stem cell derived cardiomyocytes as they recovered from that cardioplegic transport scenario. And here we're looking at the graphical representation. Basically, we found that if we look at the beating rate of the heart and normalize that to the beginning standard rate, from the time you add the transport media and put it in the cold, the cells stop beating. But then when you rewarm them and switch to normal media again, much like switching them into the body after the transplant, you see a recovery of function. And that's shown as a standard with UW only solution in the dotted black line on the left. But when we admix the ASC conditioned media, you get a marked improvement in that recovery of function as measured here by beat rate, which closely is reflecting not only beat rate per se, but also reflects or is reflected in other parameters that can be measured, such as the contraction, the degree of contraction in a longitudinal way, or various kinds of other imaging techniques, including calcium and so forth, signaling. So then we said, okay, what if we take it out to 8 hours? So here we see UW solution at 8 hours, very poor recovery shown in red comparatively. But if you admix the ASC conditioned medium or secretome, the recovery curve on the right side in green actually looks very much like the 4 hours without conditioned media. So the point is, we began to understand that perhaps we could extend our length of time out to as much as 8 hours. In the same publication, we also looked at some of the mediators of that cardiac protective effect. And we found that if we knocked out SOD3 and catalase, which are critical to dispose of reactive oxygen species, both superoxide ultimately, as well as hydrogen peroxide, we would knock out that recovery. So if we look just, for example, at panel A, just for time, this is recovery of beat rate. It's mimicked by panel B with beating velocity and C with caspase, but your UW exposure results in a very significant diminishment from 100%. ASC conditioned media with complete media, much better. SOD1 knockdown, not much change. SOD3, significant reduction, as well as with catalase, and both is even worse. So we began to understand that the activity of the secretome in this model, importantly, is mediated by the ability to enzymatically dispose of those accumulating ROS entities, which are present and actually accumulate during that cold ischemic transport. So now what we're doing is moving towards hopefully findings that can be extended into the human heart scenario. And so here we have Yuri Stukov, a wonderful cardiovascular surgeon working with me from Ukraine, a wonderful fellow working with Mark Staples, our most senior cardiac transplant surgeon. This is in our laboratory where we're working on a procedure that very thankfully we were able to learn from Jim McCulley at Harvard. He was very gracious, trained Yuri on this procedure not too long ago. And now we've got the idea that we're looking at of a pig heart ex vivo. So we took the heart actually out from the pig and maintain it pumping for an extended period of time. In fact, yesterday, Yuri just called me earlier this morning, and we were very excited. It went for 3 hours in a scenario just like this. So we really believe that this will now allow us to evaluate secretome in an immediately preclinical scenario, by which I mean the pig heart, as probably many of you know, both in terms of its vascularity and its physiology in general and its size, therefore the scalability is important, is very similar to human heart. And we've done many, many pig heart experiments over the years. And so right now we're going to be looking at secretome and other varieties of cell-based media additives in an effort to see what we can do to actually extend the length of that transplant, not only potentially in the case of donation after brain death, but potentially also after donation after circulatory death, where there has been a problem from the heart perspective, not only in terms of that external cold transport, but also in the context of the donation of the heart after circulatory death, in which it undergoes a period of warm ischemia in the donor. So basically, we're really looking at this idea of moving from cell therapy to cell secretome therapy. I want to show you about a 2-minute video here, or 3 minutes, so you get the idea of the cardiac transplant collaboration we're doing with our colleagues in Notre Dame, and then we'll close. To do this, I believe I need to share my sound, which I'll do here. Please let me know if this does not work.
For most people, the human heart beats about once every second.
Keeping time and keeping us alive.
I never thought about this until I discovered my heart was failing and I needed a new one to keep living. Now all I think about is time.
Will I get a new heart?
Will I get it in time?
And how much more time will it give me?
Clear for takeoff. Stay with me for now.
In the United States, donor hearts are considered usable only if the transit time is less than 4 hours. And every year, thousands of hearts are discarded before transplant because the transit time between the donor and the recipient is too great.
At the University of Notre Dame, engineering professor Pinar Zorlatuna is working to extend the time donated hearts can remain usable through cutting-edge cellular research.
The special thing about Professor Zorlatuna's work is that she builds the closest thing possible to a human heart in the laboratory. This allows us to test the function of our preservation solutions and optimize the time of transportation.
In collaboration with Dr. Keith March, Professor Zorlatuna has helped to identify a solution that will improve improve the condition of a donor heart and extend its preservation time from 4 to 8 hours.
Each donor heart represents a special gift, and our efforts at Notre Dame are designed to ensure that more of these gifts reach those who need them most.
I'm Eric Lewis. I'm 24 years old, and I've been waiting for my heart for 5.5 years.
I'm still waiting for a heart transplant.
I'm still waiting for my heart transplant.
The University of Notre Dame asks, what would you fight for?
Fighting for the human heart.
We are the Fighting Irish.
So that was a really cool video from Notre Dame. As you see, we collaborated closely with them, got to collaborate on that video. And if there are people, instead of from the north, but from the deeper South Florida, i.e. FIU, that want to work on this stuff with us, we'd love to collaborate with you guys. So in conclusion, I hope I've shown you some interesting examples that would let you understand a bit about adipose-derived cells and the stromal fraction of the adipose tissues and how they are readily available and have some potent pro-survival and tissue rescue effects, behaving like repair stations on the vascular highways of the body. Also, we've shared about the mediation of those effects by secretome, and I didn't have time to talk about it, but an important component of that, besides the soluble components, with another analogy in the chicken soup, it's also the chicken and the carrots and the peas, meaning the insoluble components, the extracellular vesicles or exosomes. Talked a little bit about the limiting of stroke and the importance of that idea within the context of a phase 1 clinical trial that we hope to execute eventually. Moving into some other areas, including acute lung injury of particular interest these days, and then talking about the practical cardiac transport importance. So the human ICMs showing their behavior like human hearts after exposure to conditioned media and how much that could be useful as a model to understand that biology in humans, going from the rodent heart to the human ICMs, the induced cardiomyocytes, and now also to pig hearts ex vivo, which we hope will be the stepping stone to actually move us into a clinical environment to help people have more hearts available, as you just heard in the video. This is our Heart and Vascular Hospital up here in Gainesville. And again, it's been a real pleasure to talk to all of you. I'd love to entertain any questions if that's part of the structure of the day or whatever would be best. Thank you all so much.
Thank you, Dr. March. All right, everybody hear me? Okay, good. All right, so yeah, we definitely have time for questions and discussion. If you're watching online, you can probably put them in the chat there, and we can keep an eye on that too. Chat/Q&A box. Orlando doesn't hear. Any questions from the audience? I can start with one while people are thinking. So thinking just kind of more globally, kind of a bigger picture question in terms of developing this, to scale. Can you talk a bit about the secretome, what you have to do for quality control? And you keyed in maybe at the end on things like reactive oxygen species, scavengers being important. Can you ever imagine, kind of to borrow your analogy, going from a symphony down to like a quartet, where you have some kind of controllable system with limited factors that might be feasible?
Oh man, that's a great question. Who's asking that question again now? Because I don't know the voices.
That was Josh.
Josh, that's a super idea. That's actually exactly what we're working on. If you notice the way I said it at the end, I said something like derived from or maybe informed by the cell factors. So yes, we've been looking at a defined media that we think actually will mimic in many ways, maybe not perfectly, but we're testing it to see if it mimics it to a significant enough degree that we can do something that's a little bit more like the quartet you just mentioned. I think that's a wonderful thought process. That's exactly what we hope to do, because that will be easier to generate and make. So we're working on that problem. Exactly. Good, great question.
Yeah, I have a question. Can you hear me?
Yes.
All right. So going along with your analogies of the secretome and the ASCs being kind of these repair stations on the highway, it seems that timing of these treatments and interventions is very particular and has to be done at very specific times. does then the treatments necessitate very specialized diagnostic tunings for when these things have to be employed? Because, for example, if you're carrying another analogy to use, the coffee beans versus the coffee itself, do you just give the person coffee whenever you decide they want coffee, or do they kind of let you know when they want coffee?
Yeah, that's a great question. I like the way you thought about it. You know, it's like when are they tired enough, you know, that it's actually necessary? Here we are this morning, a little bit ago. So in terms of the diagnostic aspect, we actually have been looking at the gene expressions of the hearts over time during that period of cold ischemia. And we've noticed some very specific gene alterations, transcriptomal alterations, that are concentrated importantly in some areas, mitochondrial clock or circadian rhythm, in other words, and as well inflammatory areas. And one of the things we're thinking about is looking at some of those factors to identify, can we come up with a fingerprint that reflects the degree of transcriptomal abnormality and the extent to which we can repair or at least inhibit the progression of that transcriptomal abnormality, and maybe identify prospectively which organs will do better than others, or which ones we've, say, sufficiently rescued. That's not precisely the same as the coffee thing, because that would be more like, let's wait until it gets bad and then we're going to give it some caffeine to wake up. But, you know, conceptually, trying to understand the status of what's actually happening and then respond to it in a little bit more intentional way is a great idea. Practicality will be a challenge, but, you know, we're trying.
Any other questions from the audience here? Yes, Dr. Armstrong.
Thank you, Dr. March. We appreciate your talk. I had a quick question regarding your model systems. Very fascinating with the pig hearts as well as the iPSC cardiomyocytes. My question relates to, I guess, the immune system. And I was wondering to what extent, being an allogeneic product, could the effects of the immune system be studied in these model systems?
Yeah, interestingly, we found that even crossing the xenogenic barrier, we typically have some very impressive results in almost every system we've looked at, maybe one exception or two I can think of over the years. And I think that may importantly be because a sufficiency of growth factors do in fact cross-react, you know, with their cognate receptors in the other species or in the other scenario. That's one aspect. The other that we just found out with this heart situation, and it is clear to us that different factors are more important in different organs. I mean, you can kind of think of that a little bit like the instruments are different to perform different concerts. But in terms of the enzymatic properties being important, of course, enzymes will work across species barriers because they're operating on particular chemical or molecule entities, such as in the case of the redox and the free radicals. As to the immune, though, aspects, we've seen relatively little evidence, although there are some for the cells, but in terms of secretome, very small evidence for an allo reactivity there, though I must admit we haven't had enough time to look for it. We do need to look at that. We certainly need to look when we're doing clinical studies, but that hasn't happened yet. I think that it's going to be relatively small because, of course, these are acellular, and so we're not having cells displaying, you know, the typical surface recognition molecules at all.
All right, let's thank Dr. March again. Before we let you go this morning, and we wish that you could certainly be here in person, and hopefully conditions continue to improve and we can host you here at FIU in the future. We do have a plaque that we're going to send up to you. It says, "In recognition and appreciation for your commitment to advancing cardiovascular research and your participation in our annual Miami Heart Day Symposium." And it has your name on it and our logo as well. And these were provided by our department and support from the Coulter Heart Research Foundation. So thank you again for joining us this morning.
Very welcome. Thank you for the invitation.
All right, before we move on from here, we have a few other folks that we want to recognize. So again, this day wouldn't be possible without the support of the Coulter Heart Research Foundation. We have representatives here. I think we worked very hard with both Nancy and Tracy in making this happen. And so we have a couple of plaques here for them as well. Again, in recognition of all of the work that you do for cardiovascular research throughout Florida, and thank you for helping us pull this off today, despite all the challenge. Thank you both very much.